How can the kidney produce urine that is more concentrated than plasma? How does it create a hyperosmotic medulla without using a pump that directly pushes water? The answer lies in one of the most elegant mechanisms in physiology, the countercurrent mechanism. In the next few minutes, we'll break it down step by step so it actually makes sense. First, we'll understand the anatomy involved. Then we'll explain the countercurrent multiplier in the loop of Henle. After that, we'll look at the Vasa Recta and the countercurrent exchanger. Then we'll connect everything to ADH and urine concentration. Finally, we'll see why this matters clinically. The loop of Henle has two limbs. The descending limb is permeable to water, but not to sodium. The ascending limb is impermeable to water. The thick ascending limb actively transports sodium, potassium, and chloride into the interstitium. This difference in permeability is the foundation of the countercurrent mechanism. Without it, nothing works. The thick ascending limb pumps sodium and chloride into the interstitium. Water cannot follow, so the interstitial fluid becomes hyperosmotic. Now, the descending limb is exposed to this hyperosmotic environment. Water moves out of the descending limb by osmosis. As tubular fluid flows continuously, this process repeats again and again. Small horizontal differences become large vertical gradients. This is why it is called a countercurrent multiplier. It multiplies the osmotic gradient down the medulla. The medullary gradient must be preserved. That's where the Vasa Recta comes in. It has a hairpin structure, just like the loop of Henle. As blood descends, it gains solutes and loses water. As it ascends, it loses solutes and gains water. Because of this passive exchange, the gradient is maintained. It does not wash away the medullary hyperosmolarity. This is called the countercurrent exchanger. The countercurrent mechanism creates the gradient, but ADH decides whether we use it. ADH increases water permeability in the collecting duct. So when ADH is present, water leaves the collecting duct and moves into the hyperosmotic medulla. Urine becomes concentrated. Without ADH, water stays in the tubule. Urine becomes dilute. So remember, loop creates the gradient. Vasa Recta maintains it. ADH uses it. In diabetes insipidus, ADH is absent or ineffective. So even though the gradient exists, it cannot be utilized. In loop diuretics, like furosemide, sodium reabsorption in the thick ascending limb is blocked. So the gradient cannot form properly. Understanding this mechanism makes pharmacology much easier. Let's summarize. The descending limb allows water out. The ascending limb pumps sodium out but keeps water in. This creates a hyperosmotic medulla. The Vasa Recta preserves this gradient, and ADH determines how much water is reabsorbed. If you understand this flow logically, the countercurrent mechanism becomes simple. If this explanation made the countercurrent mechanism finally click, make sure to subscribe. We're building physiology step by step, logical, exam-focused, and easy to remember. Next, we'll simplify acid-base disorders. See you in the next one.